Chapter 7, Neoplasia. In this chapter, we're going to talk about cancer. We'll discuss the epidemiology of cancer, risk factors, how they're classified. We'll go over the normal cell cycle and examine the genetic mechanism of cancer. We'll discuss the difference between benign and malignant tumors, and finally, discuss the manifestations of cancer, so what it looks like once you develop cancer. So cancer cells will first develop from some sort of a mutation in a single cell that will grow, which we say is when it proliferates. But the issue with cancer cells is that it's growing or proliferating without control. Normally, cells will grow when it is told to, but cancer cells will do so even if the signal to grow has not been initiated. They also do not die off or apoptose. And as a result, the number of cells will grow instead of staying constant. Cancer cells also tend to metastasize, especially if they are aggressive. Other terms I'd like you to be familiar with are neoplasia. Neoplasia just means a new growth, and it does imply an abnormality in that growth. And it can refer to both a benign or a malignant growth. A tumor is just a mass, so it's referring to some sort of a mass. So again, it can both be a benign or a malignant mass. A malignant tumor is basically cancer, and a benign tumor is a growth that is generally easily cured. So a little bit of epidemiology related to cancer. So it is the second leading cause of death in the U.S., second only to cardiovascular disease. And most deaths related to cancer occur in individuals over the age of 55. And between men and women, men have a 1 in 2 risk and women a 1 in 3 risk of developing cancer. The 5-year survival rate is about 66%, and that will really vary depending on the type of cancer you develop or where it develops in the body. One thing that will increase your likelihood, your prognosis, is early screening. So the earlier you detect it and the earlier you start treatment, the better your prognosis. Now, a third of cancer deaths can be attributed to lifestyle factors. So that really gives you an idea of how important it is to live a lifestyle that promotes health. So what are some of these lifestyle factors we're talking about? So one is tobacco use, so smoking. Nutrition also plays a role, so high fat, lots of alcohol intake, and obviously your nutrition can also tie into obesity. Sun exposure, the UV rays, can increase the likelihood of skin cancer. And then sexual exposure to things like HPV, which increases the likelihood of cervical cancer. Other risk factors include viruses, radiation exposure, certain chemicals like asbestos. And then if you have a compromised immune system, your body won't be able to fight off abnormal cells as well. Your genes also play a role, as well as hormones. Now, speaking of risk factors, I mentioned tobacco use. So the death rate for lung cancer has increased, and it may directly be related to smoking. And lung cancer really is important for several reasons, or one of which is the fact that it has the worst survival rate. So you really want to minimize your risk of developing lung cancer. But what's interesting is that tobacco use, although we link it really to lung cancer, it's also been shown to be related to other types of cancers. So cancers in other parts of the body, including the pancreas, the kidneys, your bladder, your mouth, esophagus, even cervical cancer. So how is cancer classified? Well, it's basically classified by the tissue or cell in which it originates. For example, if cancer is detected in brain tissue or spinal cord, we call it gliomas. If it's detected in your pigment cells, we call them melanomas. So really, where was the cancer cell first detected? So the origin is going to determine how we classify cancer. So let's briefly go over a normal cell cycle and the role of growth factors and growth factor receptors. So here we have a depiction of a normal cell, or part of a normal cell. So you can see the nucleus, we see a mitochondria here, and Golgi here. Normally, the number of cells that are produced will be equal to the number of cells that die. And so if you produce the same number of cells that died, then the total number of cells in the body will remain constant. Now, cells will divide only when they're told to do so. So this is, again, in a normal situation. And growth factors, hence it has the name growth in it, it makes sense that these growth factors are going to stimulate cell growth. And over here in this figure, the growth factors are shown in these green circles. When growth factors are released, they will bind to growth factor receptors. The receptors are shown in red, so these are the red squares. Now here I have a growth factor that's bound to a receptor, and this big yellow kind of burst just signifies that the receptor is now active. And it will activate a G protein attached to the receptor, which will turn on other enzymes within the cell. And a bunch of second messengers get turned on. Eventually, the end result is it signals this cell to grow and divide. Okay, so this is a normal process. Now let's talk about cancer and the genetic mechanisms of cancer. And we'll get back to the normal cell cycle and why that was important. So carcinogens, I'm sure you've heard of this word before. It's basically a potential cancer-causing agent. So the more carcinogens you are exposed to, your risk of cancer increases. Proto-oncogenes are normal genes. So these are genes that all of us have that code for the normal things necessary for normal proteins necessary for cell division. So in the previous slide, we talked about how we need growth factors, we need receptors, we need G proteins and enzymes, etc. for a cell to grow. Well, the genes, the code of DNA that codes to make all of those types of proteins are called your proto-oncogenes. So they include your growth factors, the growth factor receptors, all of the signaling molecules inside the cell, transcription factors, and even the genes that turn these proteins on and off. So everything involved in normal cell growth, that DNA that codes for those proteins, we call it proto-oncogenes. So this is normal, okay? Now, oncogenes are basically proto-oncogenes that have become mutated. Okay, so that code of DNA that codes for the normal things that we need for our cells to grow, once it's mutated and can potentially develop into cancer, that is now an oncogene. So they will still code for the protein that's needed for cell division, but because there is a mutation, there's going to be something wrong with it. So it could be that mutation is causing you to make too much of a particular protein, or that protein is abnormal and doesn't work exactly the way that it should. Or maybe the protein now turns on all by itself, when in a normal cell before a mutation, it had to wait for something else to tell it to turn on. Or perhaps the protein is made when it shouldn't be made in that cell, or a protein that can't turn cell division off is made, or a protein that should be made by a different cell is now being made. So there's a bunch of different ways the mutation can impact the proto-oncogene. Now, another gene I want you to be familiar with are tumor suppressor genes. Now, tumor suppressor genes will code for tumor suppressor proteins. And the name tells you essentially what it does. These are proteins that will suppress the formation of tumors. And they do that by essentially acting like a checkpoint. So they will stop cell division if it notices that the cells have mutated and is not developing properly. And as a result, the more tumor suppressor proteins you have, the less likely cancer cells are to develop. Or conversely, the lower the level of tumor suppressor proteins, then the likelihood of developing cancer will increase. So now let's go back to our cell. Now this cell is slightly different. And if you compare it to the previous one, I want you to try to see what has changed. And so if you take a look here, this cell was infected with a cancer-causing virus. In our previous normal cell slide, what you saw was growth factors being made by some other cell, and our cell in the image making growth factor receptors only. But what has happened now is this cancer-causing virus has caused our cell to also make growth factors. So basically, our proto-oncogene that codes for growth factors in this cell should be turned off. But this cancer-causing virus has now turned that proto-oncogene into an oncogene. And that mutation is now causing this cell to make growth factors when it should not. So that's what's changed. So this cell is not only making growth factor receptors, it's also making growth factors. Now, how could it promote uncontrolled cell division? Because it is essentially stimulating itself to grow. Okay? So what oncogene is to blame? It's going to be the growth factor. Proto-oncogene is now turned into an oncogene, and now that's being made. Now, if you want to stop this cell from dividing, what should you do? Well, let's try giving a drug that stops other cells from making growth factors. Well, if we stop other cells from making growth factors, those growth factors won't stimulate our cell to grow, but the problem is we're making them ourselves. So this really is not going to have much of an effect. What if we give a drug that blocks growth factor receptors? Well, if we give a drug that blocks growth factor receptors, then potentially those drugs or that chemical may bind to our growth factor receptor and sit on the activation site before our growth factors can attach. So that has the potential to work. So it could work. It can prevent the growth factors this cell is making from attaching to the receptor. What if we give a drug that inactivates growth factors in the extracellular fluid? So once our growth factor is being released, if we can inactivate these growth factors before they attach to our receptor, then that signal to grow will be reduced. So yeah, this could also potentially work because the growth factor is released into the extracellular fluid. So if we can inactivate them before it binds to the receptor, then we have a way to prevent the growth stimuli. Now here's another cell. It has the same cancer, but there's something slightly different. So take a look at what's happening here. And what I want you to notice is the growth factor and the growth factor receptor in this cell is being made in the same part of the Golgi. And so some of them are actually binding and being released already bound together. Okay? So then, would a drug that stops other cells from making growth factors work? Well, it didn't work in our previous slide. It's not going to work here either for the same reason. What about a drug that blocks growth factor receptors? In this instance, it's not going to work because our growth factor is being released already bound to the receptor. So we can't give a drug to bind to the receptor because the activation site is now already being taken up by the growth factor. What about a drug that inactivates growth factors in the extracellular fluid? Again, because they are being exocytosed already bound together, the growth stimuli has already been turned on. So essentially, no treatment that affects the extracellular, anything in the extracellular fluid, so the growth factors or the cell surface receptors, is going to be able to impact this cell. So the development of cancer is a multi-step process. So first we have some sort of initiation. So this is where the initial mutation occurs. So some sort of a mutation in our DNA. Then we have promotion. This is where the mutated cells are stimulated to divide and they start to proliferate or grow. And then we have progression. So progression is when the tumor cells are starting to now even compete with one another and to become even more aggressive as they try to compete for resources. And this is when you're going to start to see the cancerous phenotype. So you're really going to start to see the signs and symptoms and manifestations of cancer. Now, any cell can mutate. And depending on where the mutation happens, we are more likely to have malignant or benign tumors. So a normal cell, when we start off with a stem cell, has to grow up. Okay, so it has to differentiate, become different. And depending on where the mutation happens, if it's early in the differentiation process, if the mutation happens early, then we're more likely to develop malignant tumors. So here we have undifferentiated cells. So here's our stem cell. It will divide and divide and divide, etc., to eventually become a normal cell that we need. But in this process, in this tree, if the mutation happens early on, okay, where the cell is still quite undifferentiated, then these cells are more likely to become malignant. If the mutation happens later on in the process, so in well-differentiated cells, then we're likely to have benign tumors. And if you think about it, that's because cells that have differentiated, become more like the cells that they're supposed to be, then they're kind of already that type of cell. They're going to look like the cell that it needs to be. It'll start to function like it. And it's just at the way end some sort of mutation happens. So notice these cells really can work like the normal cells that they are found in, but, they are still tumors. And so that's why we have benign or working tumors. But if the mutation happens early on when the cells are still quite young, then they haven't really become who they're supposed to be. So then the mutation can really make them aggressive and really kind of change the cell. So it's going to look very different than the other cells in the tissue and also function quite differently. So benign and malignant tumors. So benign tumors, again, because the mutation happens later on in the differentiation process in well-differentiated cells or working cells, the cells will typically look like the normal tissue cells. They may even perform the normal functions of the tissue. For example, if it's an endocrine cell, it'll actually secrete the hormones that they're supposed to secrete. One issue, however, is they tend to overproduce the hormone. So you tend to have oversecretion. In benign tumors, you also typically have a capsule around the tumor, and so they don't necessarily invade neighboring tissue. And but one thing that you do have to be aware of is it's still a growth. It's still a mass, and so that growth can damage the organ that it's in by causing compression and kind of just taking up space. Malignant tumors, again, occur when the mutation happens early in the differentiation process, so in undifferentiated cells. As a result, the cells do not look like the other cells in the tissue. They also do not perform the normal functions of that organ. For example, they may secrete a hormone of some other tissue. For example, this lung malignant tumor may secrete progesterone. Okay. There's also no clear boundaries, and so they tend to send out what we call legs into surrounding tissue. And the word cancer really comes from the word crab, and it's based on the crab-like legs. Also, the idea that these cancer cells will grow and grab on and not let go like crabs. Okay. Malignant tumors will also divide rapidly. As a result, the tumors grow quickly and they will mutate faster too. So within that growth, they're not all going to be the same type of cell. Just like benign tumors, you can also get compression damage. So because, again, it's a growth, it'll compress the tissue and compress neighboring tissue as well. And commonly, malignant tumors will produce an enzyme called telomerase. So if you recall telomeres, when we talked about cell aging, were the pieces of DNA at the end of our chromosomes. And remember that every time our cells divide, the telomeres get shorter and shorter. And then once you run out of telomeres, that cell can't divide anymore. Well, telomerase replaces telomeres. So essentially, you have these cells that can divide forever because your telomeres will never get shorter. Okay. Oops, sorry. Okay, so another thing that malignant tumors do is they can metastasize. So this is when they start to spread and grow to other tissues. And a lot of the times it will travel through our bloodstream. So there are three kind of major ways metastasis occurs. So it can travel through our circulatory system or through our lymphatic system. So it makes sense that they can travel to distant tissues this way because it's kind of like the freeway of our body. It can also happen through accidental transplantation during surgery or just these cells spreading to neighbors, so adjacent organs and tissues. Now imagine you were a cancer cell. What abilities would you need to survive in the tumor? Well, the first thing to keep in mind is these are still cells, so they still need the normal things a cell needs, like food and oxygen, and they also produce waste. So in order to survive, you have to compete for your resources. As a result, only the toughest and most aggressive cells are selected for. So these are the guys that tend to survive and grow. Growth can be limited if these cells are not able to attract a new blood supply, because again, the more food and oxygen and waste you can get rid of, the more likely you're going to be able to continue to grow and multiply. What abilities would you need to metastasize? Well, the process of metastasis is actually quite difficult. Imagine a cell having to first move and then work its way in through your blood vessels into the bloodstream, survive in that really turbulent environment, that high oxygen environment with lots of white blood cells, and then it has to be able to eat its way out and make a home in a new tissue. So very few cells are successful at metastasizing, but if they are successful, it makes sense that these are going to be very aggressive cells. So Tammy has liver cancer, and her doctor did an initial molecular diagnosis and put her on targeted therapy, and the tumor shrank by almost 75%. 2 years later, the tumor was growing again, and this time it did not respond to the drug. Last year, she was diagnosed with metastasis in her femur that has not responded to that same drug either. Tammy wants to know why cancer cells stop responding. Are they not all the same cells? Well, no, they are not. So remember, tumor cells, malignant tumor cells, are prone to mutate. So when she had her initial treatment, that tumor shrank by 75%. That means the cells that remained were resistant to that treatment. So those cells continued to grow. And so using that same treatment did not affect those cells because they're growing from cells that are already resistant to that drug. Okay, so she basically needs a new treatment. Okay, a new type of treatment. All right, so we have some warning signs of cancer. And there are warning signs in adults, and since children can also get cancer, we have warning signs in children. So for adults, we have the word caution. And so some signs include a change in bowel or bladder habits, a sore that just does not heal. So if it's persistent, it doesn't heal in that normal process that it should. Unusual bleeding or discharge, a thickening or lump in the breast or anywhere else really. Indigestion or difficulty swallowing, an obvious change in warts or moles. So that's why you have to really keep track of those on your body. And a nagging cough or hoarseness. In children, we have children. C for continued unexplained weight loss. Children really should be growing and not losing weight. Headaches with vomiting in the morning. Increased swelling or persistent pain in their bones or joints. A lump or mass in the abdomen, neck, or really anywhere. Development of whitish appearance in the pupils of the eye. A recurrent fever that's not necessarily caused by an infection. Excessive bleeding or bruising. And a noticeable paleness or prolonged tiredness. So these are just some warning signs of cancer. It doesn't mean if you have a headache and you vomit in the morning, you have cancer, but just some signs to be aware of. So now let's get to what it looks like if you do have cancer. So what are the manifestations? So there are several. The first is there's going to be a change in that organ's function. So that organ will become damaged, inflammation may occur, or you'll have complete organ failure. Now, benign tumors, remember, may overproduce the normal hormones that are produced by that organ. Malignant tumors can overproduce some random hormone, okay? Number 2, we have nonspecific signs of tissue breakdown. So overall, you'll just see some protein wasting, bone breakdown. Number 3 is paraneoplastic syndrome. So paraneoplastic syndrome describes the consequences of hormones being secreted by the tumor. Okay, so let me link that to change in organ function. So let's say you have a tumor in the thyroid gland. And let's say it's a benign tumor and it's causing an overproduction of thyroid hormone. So that would describe a change in the organ function. So the thyroid gland is producing too much thyroid hormone. That's a change in the organ function. Now, paraneoplastic syndrome describes, well, all of that thyroid hormone that you're producing now, that is going to also have an effect on your body. It's going to cause certain signs and symptoms to occur. It's basically going to increase metabolism, increase protein breakdown, etc. So all of those consequences, that is described as paraneoplastic syndrome. Okay? And then we have number 4, bone marrow suppression. So your bone marrow will be suppressed. So we know we make your red blood cells, white blood cells, and platelets here. So all of those guys will reduce in number. And then you'll just have local effects of tumor growth. So bleeding in the area, and then remember it's still a growth. So we talked about compression of tissue and neighboring tissue as well. So compression of blood vessels, lymph vessels, which can lead to edema, hollow organs like your GI tract if it's near your GI, or compression of nerves that can cause pain and paralysis. And then 5, we have cancer cachexia, which is general weight loss, weakness, increased metabolic rate, loss of appetite, nausea, and vomiting. And then pain. You have this mass growing in a part of your body. It's going to be painful. And then number 7, immune system suppression. And that also goes along with the bone marrow suppression. So as you suppress the bone marrow and suppress white blood cells, your immune system is also suppressed. All right, so let's end with a few questions. So a woman has breast cancer. The doctor found the cells have very high levels of estrogen receptors. What proto-oncogene do you think was mutated in this cancer? So first of all, estrogen is a growth factor. So estrogen receptors are growth factor receptors. So what proto-oncogene was mutated? Well, she has too much estrogen receptor, so the activator or receptor gene for estrogen receptors was mutated. How could the mutated proto-oncogene have caused the cells to grow? Well, estrogen, again, is a breast tissue growth stimulator. So the more estrogen receptors, the greater the growth signal. Now, how would each of the following therapies work on this cancer? So tamoxifen. This is an anti-estrogen drug. So this person has too much estrogen receptors. If you give a drug that destroys estrogen, then it can reduce the growth signal. So that might help. What about antibody that is directed to the tumor cell that has a toxin attached to it and it can deliver it to the tumor? Well, that would be a great way because it's targeted therapy. So this antibody is going to target the tumor cell, deliver the toxin, and destroy the tumor. So that's how it's going to work. And then we have a lumpectomy followed by local radiation. So lumpectomy is removal of the cancer tissue. So you're going to remove the tissue from the body. So that's one way to get rid of it. And you will always follow that by local radiation to ensure that you didn't miss anything. And local radiation is local, so it's not systemic, and it's going to cause lethal mutations in cells that are in the S and M phase of the cell cycle. So essentially, cells that are rapidly dividing are more likely to be in that phase, so they're more likely to be affected by the radiation. Now, a second woman has breast cancer, but her tumor cells have normal levels of estrogen receptors. Instead, they have G proteins that are constantly active, producing high levels of cyclic AMP. How could this cause the cells to grow? So if you recall, when our growth factor bound to our growth factor receptor, it caused G proteins to turn on and other enzymes and second messengers. So in this individual, the estrogen is normal, the estrogen receptor is normal, but that G protein that should normally only be turned on when estrogen binds to the estrogen receptor is now always on. So if it's always on, essentially you're always telling the cell to grow, even though a signal, the signal, essentially estrogen binding to the receptor, didn't even have to happen, okay? Which of the 3 treatments in the previous slide, tamoxifen, which was our anti-estrogen drug, or the antibody that was targeted to the tumor cell delivering a toxin, or the lumpectomy followed by local radiation, which one of these would probably not work for this client? Well, if you do a lumpectomy and remove the tissue and follow it with radiation, you're still removing the tissue, so that'll help. If you give an antibody with a toxin, you're delivering it to the tumor, so that will also help. The one that won't help is the tamoxifen, because tamoxifen reduces estrogen. Remember, in this woman, the G protein is on whether or not estrogen binds to the receptor. So getting rid of estrogen won't really do anything for her. Okay, so the tamoxifen is but will not work. This client's problem doesn't seem to be increased activity of the estrogen receptor, so reducing estrogen will have no effect. All right, so that is our chapter on cancer. Thank you.